So my name is Jay. I am a PhD student in Dr. Joyshri Kalpathy Kramer's lab at the Martino Center, and I'll be giving a talk on fMRI task-based variance across subjects. So to start, I want to give a brief introduction to fMRI. fMRI is a method developed to capture regional and uh, time varying changes in brain metabolism. And this is due to the fact that when neurons get activated, there's an increased flow of oxygenated blood to the area. And this change in blood oxygen level dependent contrast can be detected via MRI. Um, there's a few issues with this bold contrast that I want to quickly mention. And that's the first issue is that these detected signal changes are very small, only an absolute difference of 1 to 2%. And that wouldn't necessarily be a problem if not for the fact that noise can in fact be larger than the signal of interest. And there's a lot of sources of noise. So you have system noise, you have cardiac and respiratory induced physiologic noise, you have motion effects, which cause the partial volume effects we're seeing, and you just have a general variability and bold contrast due to baseline neural metabolism, which means that your caffeine intake for the day, the time of day itself, what the ambient settings are like, and even different regions of the brain having different absolute bold signal changes. So with all that in mind, how do you separate the true signal from the noise? And you do that by getting repeated measurements. We expect that the noise is uncorrelated, whereas the true signal is correlated with the task of interest. And so by getting many, many repeated samplings, we can isolate the true signal. So here's an example of a common block-based fMRI experiment. You have a block, which is a single contiguous occurrence of the task or stimulus. The task is usually some visual or auditory or motor-based task. And then you have this alternating sequence of a block followed by a rest period, block followed by a rest period. And so what we're gonna assess over the course of this talk is across block variance. So how much variance in signal do you get in this single contiguous occurrence of the task? Across run variance, so across this entire alternating section, how much variance do you see? And then across session variance, so a session being when a patient enters and then finally exits the scanner. So let's start with the most broadest one of those, which is across session variance. And we're going to look at a case study done a long time ago, back in 1999, actually. And in this study, they scanned a single subject a total of 99 separate times. And they did 33 sessions for a motor task, 33 for a visual task, and 33 for a cognitive task. And the general point that they wanted to ask was, is there significant across session variance? And if there is significant across session variance, does that necessarily invalidate the results that might be obtained by a single session? And they did their best to control for any artificial between session differences. So they always had the same operator controlling the scanner. They always used the same amount of ambient light and sound levels. The instructions were always identical, etc. And in general, what they found is that there's a significant amount of cross-session variance. What you can see on this figure is that these spots right here are significantly activated voxels during the specific task after multiple comparisons correction. And what you can notice is that there's a large variability in the spatial distribution of these activated voxels. For example, in some of these cases, you can see like hundreds of activated voxels, whereas other cases like session 30 and session number 4, there were 0 activated voxels found. So to more rigorously assess the effect of this, they used fixed effects and random effects modeling. I'll skip over some of this part, but I think the more important and intuitive part of their results was this graph right here. So what you can see here is that most of these voxels demonstrated low within session variance. So that's determined by the fact that these error bars relative to the signal are relatively small. But there's a high across session variance. So this one voxel, in some cases, is significantly activated. In other cases, it's baseline. Secondly, the case you actually want to see is when there's low within session variance and low across session variance. But what the authors find is that more likely you're going to find voxels like this, where there's significant across session variance. Now, that was back in 1999. And so the question is now, has this issue been resolved? And really, current literature strongly suggests that it has not. And we're going to look at a few of the more recent papers in the field. This is a 2017 paper where they assess not only the cross-section variance, but also the across runs and across blocks variance, as well as just the general modeling measurement error variance that we see. This is a rather busy figure, so I'll try to focus your attention on the main point from here. But what they wanted to get out was that the amount of variance is not spatially homogeneous. So what you can see is that there's a lot more variance in the pixels at the edges of the brain and also in the ventricle region, signifying that there's probably some motion-based artifacts and some physiologic noise as well. Secondly, they wanted to mention that the across blocks variance is relatively low, as you can see, which makes sense because the temporal difference in a block is much smaller than the temporal change in a run or a session. To further quantify this idea of spatial inhomogeneity of variance, they looked at which form of variance was most significant in different regions of the brain. And what they found was that the occipital cortex was most likely to be to have the highest across session variance, whereas most of the gray matter had the highest across run variance. And their conclusion was that based on the type of task you're doing, you should change your experiment setting. So if you know you want to stimulate the occipital cortex, it might be better to have fewer sessions so that you don't run into this issue of a cross-session variance. And the next thing that they did was they looked at network analysis. So instead of just looking at these individual voxels and isolations, we know that there's some sort of connectivity that's induced by these different tasks. And so what they found was that so what I want you to notice about this is these are each different networks. And what I want you to notice is that the measurement error is the largest in most cases. So you can see this white pie chart block is 50 to 75% of most of the error present across networks. But what they also notice is that the cross block variance, while it's negligible for somatosensory networks, it's not negligible for higher order cognitive networks. For example, this being an emotion task. So if you're showing people pictures of faces. And so the real takeaway from this study was that different regions and different tasks exhibit different variance patterns. And to generate, useful experiments, you need to account for these differences. The next paper I wanted to mention was a paper that looked at what they termed analytic degrees of freedom. And that's the idea that given the same set of data, how many different types of results can you get from teams using different processing methods? Now, processing for fMRI is a very nuanced approach, and there's going to be some difference in how teams do it, but how much of a difference does this make? And so they gave 70 teams, 69 of whom who had previous fMRI publications, the same set of data. And because these teams all have previous fMRI publications, it's expected that they know what they're doing, essentially. However, the tasks that were performed show ambiguous agreement at best. So if you look at the data that these authors showed, you want this column to either be close to 0 or close to 1. So in this case, most teams found non-significant findings, which is good. They're all in agreement. Similarly, here, most teams found significant findings for this task. Once again, good. But you can see some of these other hypotheses have low agreement. In this case, essentially 1/3 of teams had a different hypothesis setting. And so what is the source of this agreement? And what they find is that the unthresholded maps were strongly correlated with each other, but the binary inference decisions were not. So what you see here is that most teams fall within this main cluster, and the unthresholded probability masks were highly correlated with each other. But after each team applied their own processing pipelines, you get very different inference decisions as seen by these red and green blocks up here. And what the authors note is that the analysis of the underlying statistical maps, so these highly correlated maps, if you apply the same processing pipeline to them, you get very similar binary inference decisions. But the fact that each team got different inference decisions means that differences in spatial smoothing, analysis software, and how each team did statistical corrections for multiple comparisons highly affects the decision, the inference decision. And the final paper I wanted to look at was a very recent paper from Duke University on the test-retest reliability of fMRI. And really, this study just tests, is there a strong ICC between repeated measurements for patients on fMRI, tasks? And so what they did was they found 45 patients and scanned them twice, 140 days apart on average, which is a relatively long time, I should mention, but the results speak for themselves, which is that while the group level activations showed strong activation in the correct regions, the ICC between test-retest was extremely poor. What I mean by this is if you look at this figure, you can see that for the emotion task, there's activation, group level activation in the amygdala, which is what we expect. But then if you look at the ICC per patient, the ICC in the amygdala region is close, it's below 0.4 because it's not colored on this scale. So extremely poor intra-class correlation coefficient. Furthermore, they wanted to check if there was any agreement of ICC across regions. So for once again, if we look at this emotion task, not even looking at just the amygdala, which is the region of interest, but looking at the other regions in the brain, you see that across regions and across tasks, there's poor ICC, less than 0.4, and very few of these regions break 0.5 ICC. As a sanity check, they did get the ICC for structural measurements. So this is cortical thickness, this is surface area, and as expected, there's very high ICC for these measures. It's just the fMRI results that have poor ICC they found. The solutions that this paper posed were relatively straightforward. It's just you need more data per subject and more subjects per study. And what they state is that the vast majority of these fMRI studies that are published are extremely underpowered. I just want to end with a quick rebuttal. So this Duke paper got a lot of press uh, in recent times, and so 3 days ago, a group published a short rebuttal to this paper, and their rebuttal is simply that fMRI can be quite reliable when done properly, and they state that the small sample size used in the Duke study, so 45 patients, the task types that they used and the dated ROI measures led to negative results, but that modern fMRI research can be done properly. But just to conclude, these issues regarding reliability of fMRI have been around for a long time and are still being brought up in present day. So yeah, I'd like to finish with that, and if there are any questions.